In a sterile laboratory somewhere in the world right now, scientists are working with pathogens so dangerous that a single mistake could change everything. We're talking about engineered viruses that make COVID-19 look like a common cold, bacteria designed to resist every antibiotic we have, biological weapons that exist purely in theory, except they don't. They're real, they're being studied, and the safety protocols keeping them contained aren't as foolproof as you might hope. What happens when one of these lab-created nightmares escapes? I'm talking about pathogens that don't follow nature's rules. Organisms that have been modified, enhanced, weaponized in ways that evolution never intended. And before you think this is some distant science fiction scenario, let me tell you something that'll keep you awake tonight. There have been over 200 documented laboratory accidents involving dangerous pathogens in the last decade alone. 200 that we know about. But here's what makes this different from every other pandemic discussion you've heard. We're not talking about natural spillover events or random mutations. We're talking about intentionally designed biological weapons that could make every pandemic in human history look like a minor inconvenience. And the people creating them? They're not movie villains in underground bunkers. They're respected researchers in government labs, university facilities, and private companies around the world. Let me start with something that happened just 3 years ago that barely made the news. A laboratory in Atlanta accidentally mixed 2 different viral samples, creating a hybrid pathogen that shouldn't exist. The mistake was caught within hours. The samples destroyed. But for those few hours, something new walked the earth. Something that combined the transmissibility of one virus with the lethality of another. The researchers involved were sworn to secrecy. The incident was classified, but it happened. And that was just an accident? The world of pathogen research exists in a shadow realm between legitimate science and potential catastrophe. It's called dual-use research of concern, and it's exactly what it sounds like. Research that has legitimate medical applications but could also be weaponized. The same techniques used to develop life-saving vaccines can be used to create unstoppable plagues. The line between protection and destruction is thinner than a laboratory glass slide. Here's how these engineered pathogens are actually created and why they're infinitely more dangerous than anything nature produces on its own. Natural pathogens, even the worst ones in history, have limitations. They evolved to balance transmission with virulence because killing the host too quickly limits spread. It's evolutionary mathematics. But lab-created pathogens don't follow these rules. Scientists can engineer viruses that are both highly transmissible and extremely lethal because they're not constrained by natural selection pressures. The process starts with something called gain-of-function research. Scientists take an existing pathogen and modify it to make it more dangerous, more transmissible, more resistant to treatment, more capable of evading immune systems. The official justification is that by understanding how these enhanced pathogens work, we can better prepare defenses against them. The unofficial reality is that we're creating weapons that didn't exist before. Take the 2005 reconstruction of the 1918 flu pandemic virus. Scientists literally brought back one of history's deadliest pathogens from tissue samples and genetic sequences. They published the complete genetic code online, essentially giving anyone with basic laboratory skills the blueprint to recreate Spanish flu. The justification was research. The result was that one of humanity's worst killers was back from extinction and available to anyone with internet access and a basic understanding of molecular biology. But that's just the beginning. Modern techniques go far beyond simple reconstruction. CRISPR gene editing allows scientists to make precise modifications to pathogen DNA with unprecedented accuracy. They can remove the genes that limit transmission, enhance the genes that increase virulence, and add entirely new capabilities that never existed in nature. We're talking about viruses that can survive on surfaces for weeks, bacteria that produce multiple toxins simultaneously, and pathogens that can evade detection by every diagnostic test we have. The most terrifying part isn't what these pathogens can do individually. It's what happens when you combine multiple enhancements in a single organism. Imagine a virus with the transmission rate of measles, the case fatality rate of Ebola, and the ability to remain asymptomatic for weeks while spreading. This isn't science fiction. These are real modifications that researchers are making right now in laboratories across the globe. And the safety systems designed to contain them? They're failing more often than anyone wants to admit. Laboratory biosafety levels range from BSL-1 to BSL-4, with BSL-4 being the highest security level reserved for the most dangerous pathogens. These facilities are supposed to be impenetrable fortresses of containment. Multiple layers of security, positive pressure systems, specialized ventilation, trained personnel in full-body protective suits. But even BSL-4 labs have accidents. In 2014, vials of smallpox were found forgotten in a storage room at the CDC, not in a BSL-4 facility, but in a regular storage area where anyone could have accessed them. These vials had been sitting there for decades, completely outside any containment protocol. If something that simple can happen with smallpox, what else is sitting unguarded in laboratories around the world? The human factor is always the weakest link. Researchers working 16-hour days under intense pressure, budget cuts leading to deferred maintenance on critical safety systems. Personnel who become complacent after years of handling dangerous materials without incident. Every one of these factors increases the probability of the unthinkable happening. But here's what really keeps experts awake at night. It's not just accidental release they're worried about. The knowledge and techniques for creating enhanced pathogens are spreading faster than our ability to control them. What used to require a team of PhD researchers and millions of dollars in equipment can now be done by a graduate student with access to standard laboratory equipment. The democratization of biotechnology means that the power to create world-ending plagues is increasingly available to anyone with sufficient motivation and basic skills. We're living through a moment in history where the barrier to creating catastrophic biological weapons is lower than it's ever been, while the potential consequences are higher than ever before. And the systems we have in place to detect and respond to these threats? They were designed for natural pandemics, not engineered ones. When COVID-19 emerged, it took months to understand its basic characteristics, how it spread, who was most vulnerable, what treatments might work. With a natural pathogen, this timeline is frustrating but manageable. With an engineered pathogen designed to evade our countermeasures, those months could be the difference between containment and global catastrophe. The detection systems we rely on are based on recognizing known patterns, but engineered pathogens can be designed to avoid these patterns entirely. They can be created to produce symptoms that mimic common illnesses, to evade standard diagnostic tests, to remain undetectable until they've already spread extensively. By the time we realize what we're dealing with, it could be far too late to stop it. And that's assuming we even recognize that we're dealing with an engineered pathogen at all. How do you tell the difference between a natural mutation and deliberate modification? The techniques used to create enhanced pathogens often leave no obvious fingerprints. An engineered virus can look entirely natural until you examine its genome with sophisticated analysis that most diagnostic laboratories simply don't have the capability to perform. This brings us to perhaps the most chilling aspect of this entire scenario. The people creating these enhanced pathogens often have no idea how dangerous their creations actually are until it's too late. They're working with theoretical models and computer simulations, but the real-world behavior of these organisms can be completely different from predictions. Consider what happened with the mousepox experiments in Australia in 2001. Researchers were trying to create a contraceptive vaccine for mice by modifying mousepox virus. Instead, they accidentally created a virus so lethal that it killed even vaccinated mice that should have been immune. The modified virus was 100% fatal in their test subjects. What should have been a mild infection became an unstoppable killer through a single genetic modification that no one predicted would have that effect. If this kind of unexpected enhancement can happen with mice, what happens when similar modifications are made to pathogens that affect humans? The researchers involved in the mousepox experiment were horrified by their results. But by then, the knowledge existed. The techniques had been demonstrated. The blueprint for accidentally creating unstoppable pathogens was out there for anyone to follow. This is the world we're living in right now. Laboratories around the globe are conducting research that could accidentally or intentionally create pathogens far more dangerous than anything in human history. The safety systems designed to contain them are imperfect and failing with increasing frequency. The knowledge needed to create these threats is spreading faster than our ability to control it. And if one of these engineered nightmares does escape, whether through accident, negligence, or malicious intent, our current systems for detecting and responding to biological threats are completely inadequate for the challenge we would face. The question isn't whether this scenario is possible. The question is whether we're prepared for when it happens. And based on everything we know about laboratory safety, pathogen research, and our pandemic response capabilities, the answer should terrify you. The reality of our preparedness becomes even more disturbing when you examine what happened during the early days of COVID-19. Remember those first crucial weeks when the virus was spreading silently through communities while health officials insisted the risk was low? That was with a naturally occurring pathogen that followed predictable patterns. Now, imagine the same scenario, but instead of a virus that evolved over time, we're dealing with something designed in a laboratory to exploit every weakness in our detection systems. Here's what that timeline would actually look like. Day 0. A laboratory accident occurs. Maybe it's a researcher who cuts themselves while handling a sample. Maybe it's a containment system that fails during routine maintenance. Maybe it's something as simple as a labeled vial being placed in the wrong storage unit. The engineered pathogen escapes its controlled environment and begins spreading. But here's the critical difference. This isn't like natural spillover events where we can trace patient 0 to a specific location or exposure. Engineered pathogens can be designed with incubation periods that stretch for weeks or even months. The initial infection could travel across continents before the first symptoms ever appear. Days 1 to 30. The pathogen spreads silently. Infected individuals feel perfectly healthy while unknowingly transmitting the organism to everyone they encounter. Airport terminals, subway systems, office buildings, schools. Each infected person becomes a super spreader without realizing it. The pathogen, designed to evade immune responses, multiplies unchecked in human hosts. Traditional surveillance systems detect nothing because they're looking for known signatures. The engineered pathogen presents as ordinary respiratory symptoms, if it presents symptoms at all during this phase. Emergency rooms see a few more cases of what looks like seasonal flu. Nothing triggers alarm bells because the pathogen was designed not to trigger them. Days 30 to 60. The first severe cases begin appearing. But they don't cluster around a single geographic location like natural outbreaks. They appear simultaneously in major population centers across multiple continents: New York, London, Tokyo, Sao Paulo, all reporting similar cases at the same time, which should be impossible with a natural pathogen, but is exactly what you'd expect from something that's been spreading undetected for weeks. Health officials are confused. The epidemiological patterns don't make sense. There's no clear source, no obvious transmission pathway, no animal reservoir to investigate. The cases seem to appear spontaneously, which delays recognition that we're dealing with a coordinated outbreak rather than isolated incidents. By the time someone realizes these cases are connected, the pathogen has already established itself in dozens of countries. Millions of people have been exposed. The window for containment has closed without anyone knowing it was ever open. Days 60 to 90. The engineered nature of the pathogen becomes apparent as it demonstrates capabilities that violate everything we know about natural evolution. It resists every antiviral treatment in our arsenal because it was designed to resist them. It evades diagnostic tests because it was engineered to be invisible to standard detection methods. It shows unusual stability in environmental conditions because someone modified it to survive longer outside human hosts. This is when panic sets in, not just among the general population, but among the scientific community itself. Researchers worldwide begin analyzing samples, trying to understand what they're dealing with. The genetic analysis reveals modifications that couldn't have occurred naturally. Someone created this pathogen intentionally. But identifying it as engineered doesn't help us stop it. In fact, it makes the situation worse because now we know we're fighting something designed to defeat our countermeasures. Every strategy in our pandemic playbook was developed for natural pathogens. None of them account for organisms specifically engineered to exploit weaknesses in our response systems. Days 90 to 180. Global health systems begin collapsing under the strain. Hospitals overflow with patients suffering from a disease that doesn't respond to any known treatment. Healthcare workers, despite protective equipment, continue falling ill because the pathogen was designed to overcome standard precautions. Supply chains break down as entire regions go into lockdown simultaneously. Unlike natural pandemics that spread gradually, giving communities time to adapt, this engineered threat hits everywhere at once. There's no safe haven, no unaffected region that can provide assistance or resources. Economic systems that survived COVID-19 collapse entirely. The stock market doesn't just crash, it ceases to function as markets close indefinitely. Currency becomes worthless when the systems maintaining its value shut down. International trade stops when ports close and transportation networks fail. But here's where it gets truly horrifying. This scenario assumes the escaped pathogen was created for legitimate research purposes and only accidentally became a global threat. What if it wasn't an accident? What if someone deliberately created and released an engineered pathogen designed specifically to cause maximum damage to human civilization? The techniques exist right now to create pathogens that target specific genetic markers. Meaning you could theoretically design a virus that affects certain populations while leaving others relatively unharmed. You could engineer bacteria that remain dormant until activated by specific environmental triggers, allowing for delayed or coordinated attacks. You could create self-limiting pathogens that burn themselves out after achieving specific objectives, leaving infrastructure intact for whoever survives. These aren't theoretical possibilities. These are real capabilities that exist in laboratories today, and the knowledge needed to use them is spreading through published research, academic conferences, and online communities faster than any regulatory framework can possibly control. The most terrifying aspect of intentional biological warfare isn't the immediate casualties, it's the complete breakdown of trust in scientific institutions that would follow. How do you rebuild society when people can never be certain whether the next outbreak is natural or artificial? How do you maintain international cooperation when any nation could be harboring secret bioweapons programs? Even the suspicion of engineered pathogens changes everything. During a natural pandemic, nations work together because they face a common enemy. But when the threat might be artificial, when it might have been created by human hands for human purposes, cooperation becomes nearly impossible. Everyone becomes a potential suspect. Every laboratory becomes a potential weapons facility. This paranoia would extend far beyond government levels. Communities would turn inward, rejecting outsiders who might be carriers of unknown threats. Scientific research itself would become suspect as people question whether researchers are trying to protect humanity or develop new ways to destroy it. The very institutions we depend on to develop treatments and vaccines would lose public trust at exactly the moment when we need them most. And here's the cruel irony. The more we research dangerous pathogens in order to defend against them, the more we increase the risk of creating or accidentally releasing them. It's a paradox with no clear solution. We need to understand these threats to protect ourselves, but the act of studying them creates the very dangers we're trying to prevent. This is the world we've created through decades of advancing biotechnology without adequate consideration of the consequences. We've built the tools to engineer life itself, but we haven't built the wisdom to use them responsibly. We've democratized the power to create and modify living organisms, but we haven't democratized the understanding of what that power means. The escape of a lab-created pathogen isn't just a medical emergency. It's an existential threat to human civilization as we know it. And based on current trends in laboratory safety, research proliferation, and global preparedness, it's not a question of if this will happen. It's a question of when. The timeline I just described assumes we'd even recognize we were under attack. But what if we didn't? What if the pathogen was designed to look like multiple different natural outbreaks occurring simultaneously around the world? This is where the sophistication of modern bioweapons research becomes truly diabolical. Scientists have already demonstrated the ability to create what they call chimeric pathogens, organisms that combine genetic material from multiple different sources. Imagine a single engineered organism that presents as seasonal flu in some patients, pneumonia in others, and something resembling food poisoning in a third group. To health officials tracking these outbreaks, they would appear to be dealing with three separate, unrelated problems. Resources would be divided, response efforts would be scattered. No one would realize they were fighting a single engineered threat until it was far too late to coordinate an effective defense. The research proving this is possible already exists. In 2016, scientists demonstrated that they could create a virus that would cause different symptoms depending on the genetic background of the infected individual. The same pathogen would present as a mild respiratory illness in people with certain genetic markers, while causing severe neurological symptoms in others. The implications are staggering. You could engineer a pathogen that appears to be a minor outbreak among one population while simultaneously devastating another. International health organizations would focus their attention on the severe cases while the minor outbreak spreads uncontrolled, eventually revealing itself as part of the same engineered threat. But even this level of sophistication pales in comparison to what's theoretically possible with current technology. Scientists are now working on what they call temporal pathogens, organisms designed to change their behavior over time. Picture this scenario. A pathogen is released that initially behaves like a common cold, highly transmissible but with minimal symptoms. Health authorities barely notice it because it seems harmless. People continue their normal activities, traveling, working, socializing, unknowingly spreading this harmless virus across the globe. Then, after a predetermined period, maybe 6 months, maybe a year, the pathogen activates dormant genetic sequences. Suddenly, this harmless cold virus transforms into something lethal. People who were infected months earlier and have completely recovered begin falling seriously ill. The activation happens simultaneously worldwide because it's controlled by an internal genetic timer, not by external conditions. By the time anyone realizes what's happening, virtually everyone on Earth has been exposed to the dormant form of the pathogen. There's nowhere to run, nowhere to hide, no uninfected population to draw assistance from. The entire human species becomes a victim simultaneously. This isn't science fiction. The genetic switches needed to create temporal pathogens already exist in nature. Researchers have identified them, studied them, and learned how to manipulate them. The technology to create a pathogen with delayed activation is available right now in laboratories around the world. And here's what makes this scenario particularly terrifying. Our entire pandemic response system is built on the assumption that we'll have time to react. We track emerging threats, develop countermeasures and implement responses based on observing how a pathogen behaves over time. But with temporal pathogens, there is no time to react. By the time you know what you're dealing with, the opportunity to respond has already passed. Even our most advanced diagnostic systems would be useless against this type of threat. You can't test for something when you don't know it exists. You can't develop treatments for a pathogen that appears harmless until it's too late to treat anyone. The people creating these capabilities aren't cartoon villains plotting world destruction in hidden lairs. They're researchers at prestigious universities and government laboratories, publishing their findings in respected scientific journals. They present their work at international conferences where the audience applauds breakthroughs in genetic engineering and synthetic biology. The disconnect is breathtaking. The same techniques being celebrated as revolutionary advances in medicine are also the foundation for creating weapons that could end human civilization. The researchers developing these technologies often seem genuinely oblivious to the dual-use implications of their work. I've spoken with scientists who've made significant contributions to gain-of-function research, and many of them express surprise when asked about the weapons potential of their discoveries. They're so focused on the legitimate medical applications that they haven't fully considered what happens when their techniques fall into the wrong hands. This institutional blindness is perhaps our greatest vulnerability. We're advancing the science of pathogen engineering faster than we're developing the wisdom to control it. Every breakthrough brings us closer to capabilities that could destroy us, but also closer to treatments that could save us. It's a race between salvation and catastrophe, and we have no way of knowing which will reach the finish line first. The regulatory frameworks meant to oversee this research are hopelessly inadequate for the challenge. Most oversight committees evaluate research proposals based on their intended purpose, not their potential for misuse. A proposal to develop vaccines against bioterrorism threats sounds perfectly reasonable until you realize that the same research will also teach people exactly how to create those bioterrorism threats. International agreements on biological weapons are even more inadequate. The Biological Weapons Convention, signed by most nations, prohibits the development of offensive biological weapons, but it doesn't prohibit defensive research, and the line between defensive and offensive capabilities has become meaningless in the age of synthetic biology. Any research that helps us defend against biological weapons also teaches us how to create them. Any technique for detecting engineered pathogens also reveals the methods needed to make them undetectable. Every advance in medical countermeasures simultaneously advances weapons development. The result is a world where biological weapons capabilities are proliferating under the guise of legitimate medical research, protected by international agreements that were written before these dual-use technologies existed. And the proliferation isn't limited to major powers with sophisticated military research programs. The democratization of biotechnology means that smaller nations, non-state actors, and even individuals can access the tools needed to create devastating biological weapons. A terrorist organization doesn't need to steal nuclear material or recruit rocket scientists anymore. They need access to a basic laboratory and someone with a graduate degree in molecular biology. The barriers to entry for biological terrorism have collapsed, while the potential for catastrophic damage has expanded exponentially. Consider what a dedicated group with modest resources could accomplish today. They could acquire pathogen samples from laboratory supply companies that sell to legitimate researchers. They could download genetic sequences from public databases maintained by health organizations. They could purchase the equipment needed for genetic modification from the same companies that supply university laboratories. With these readily available materials, a small team could potentially create biological weapons more devastating than anything in national military arsenals. And they could do it without triggering any of the monitoring systems designed to detect weapons proliferation because they're using the same materials and methods as legitimate researchers. The invisibility of biological weapons development makes detection and prevention nearly impossible. You can't monitor biological weapons programs the way you can monitor nuclear programs. because they use the same facilities, equipment, and expertise as legitimate medical research. A nation could be developing the most sophisticated biological weapons arsenal in human history while simultaneously hosting international conferences on pandemic preparedness. The same scientists, the same laboratories, the same equipment would be involved in both efforts. The only difference would be intent, and intent is impossible to verify from outside observation. This dual-use problem extends beyond just the creation of weapons. Even our efforts to defend against biological attacks can be turned against us. Research into how to detect engineered pathogens reveals exactly what signatures to avoid when creating them. Studies on how biological weapons might be deployed provide detailed blueprints for deployment strategies. We're trapped in a paradox where every step we take to protect ourselves also provides a roadmap for attacking us. The more we learn about biological threats, the more vulnerable we become to those same threats. The more sophisticated our defenses become, the more sophisticated the weapons designed to overcome them will be. And underlying all of this is a fundamental question that nobody wants to address directly. Do we actually want to stop biological weapons development? Or do we just want to control who has access to them? The major powers investing billions in defensive biological research aren't just preparing to defend against attacks. They're also maintaining the capability to launch them. The infrastructure, expertise, and knowledge base needed for defense is identical to what's needed for offense. This creates a security dilemma where every nation's efforts to protect itself appear threatening to others. When one country develops better detection systems for biological weapons, other countries assume they're also developing better ways to evade detection. When a nation builds high-security laboratories for studying dangerous pathogens, neighbors wonder whether they're really studying them or weaponizing them. The result is an arms race conducted in secret, where progress is measured not by public announcements, but by the sophistication of threats we're supposedly defending against. Each side assumes the others are further ahead than they appear, leading to acceleration in research that nobody wants to acknowledge. Meanwhile, the actual likelihood of a biological weapons attack or laboratory accident continues to increase with each passing year. More laboratories, more research, more dangerous pathogens, more people with access to dual-use technologies. The mathematics of risk are working against us, and our safety systems aren't keeping pace with the expanding threat. We're conducting a global experiment in biological risk management, and we won't know the results until it's too late to change them. The mathematics of this risk become clearer when you examine what's happening in laboratories right now behind closed doors where cameras aren't allowed and journalists aren't welcome. Because while we debate the theoretical possibilities of biological weapons, researchers are already crossing lines that would have been unthinkable just a decade ago. In 2019, a team of scientists successfully created a synthetic version of horsepox virus from scratch, using nothing but commercially available DNA sequences and standard laboratory equipment. Horsepox is closely related to smallpox, and the techniques used to create one can easily be adapted to create the other. The researchers published their methods in complete detail, essentially providing a cookbook for recreating one of history's most devastating diseases. The justification was that this research could lead to better smallpox vaccines. The reality is that they demonstrated how to bring extinct pathogens back to life using technology available to anyone with sufficient funding and basic laboratory skills. They proved that eradication of a disease doesn't mean it's gone forever if someone knows how to rebuild it from genetic blueprints. But synthetic reconstruction is just the beginning. The real breakthrough came with something called directed evolution, a technique that allows researchers to force pathogens to evolve specific traits in laboratory conditions. Instead of waiting for natural selection to produce desired characteristics, scientists can create selective pressures that drive evolution in predetermined directions. Here's how it works. Researchers take a pathogen and expose it to conditions that favor certain traits while eliminating others. Want a virus that's more resistant to heat? Gradually increase laboratory temperatures while allowing only the most heat-resistant variants to survive and reproduce. Want bacteria that can evade a specific antibiotic? Expose successive generations to increasing concentrations of that drug until only the resistant strains remain. This process can be repeated indefinitely, with each generation becoming more adapted to whatever conditions the researchers choose to impose. The result is evolution at hyperspeed, producing organisms with characteristics that would take centuries or millennia to develop naturally. The technique has already been used to create influenza viruses that can spread more easily between mammals, anthrax bacteria that resist multiple antibiotics simultaneously, and coronaviruses that can infect species they've never encountered before. Each of these achievements was presented as important medical research, but the same methods can be used to create biological weapons more sophisticated than anything natural evolution has produced. What makes directed evolution particularly dangerous is its unpredictability. Researchers can select for specific traits, but they can't always predict what other characteristics will emerge alongside the desired ones. A virus evolved for greater transmissibility might spontaneously develop increased virulence. Bacteria selected for antibiotic resistance might acquire new toxin-producing capabilities. These unexpected developments aren't theoretical concerns. They're documented realities that have occurred repeatedly in directed evolution experiments. Researchers consistently report being surprised by the unintended consequences of their work, yet they continue pushing forward with increasingly ambitious projects. The pace of this research is accelerating exponentially. What used to require months of careful experimentation can now be accomplished in days using automated systems and artificial intelligence. Computer algorithms can design genetic modifications that would have taken teams of researchers years to conceptualize. Laboratory robots can implement those modifications without human intervention, testing thousands of variants simultaneously. We're approaching a point where the creation of novel pathogens will be limited only by imagination, not by technical capability. Any characteristic that can be described can potentially be engineered into a living organism. Any combination of traits that can be conceptualized can be tested and refined through directed evolution. But here's what keeps me awake at night. It's not just the sophisticated research happening in high-security government facilities. It's the undergraduate students who are learning these techniques as part of their standard curriculum. Synthetic biology has become so mainstream that it's now taught in introductory college courses. Students who've never taken an advanced biology class are learning to engineer genetic circuits, modify organism behavior, and create novel biological functions. What used to be cutting-edge research requiring PhD-level expertise is now considered basic education for anyone pursuing a career in the life sciences. These students are graduating with skills that would have been classified as weapons-related knowledge just 20 years ago. They're entering a job market where biotech companies actively recruit people with experience in genetic engineering and synthetic biology. They're starting their own companies, publishing their own research, and pushing the boundaries of what's possible with living systems. The knowledge is spreading faster than our ability to track it, regulate it, or control it. Every research publication, every conference presentation, every online tutorial contributes to a growing body of information about how to manipulate life at the molecular level. This information doesn't disappear once it's shared. It accumulates, builds upon itself, and becomes accessible to anyone with curiosity and internet access. Consider what happened with CRISPR gene editing technology. When it was first developed, it was confined to a handful of specialized laboratories with the expertise and equipment needed to use it effectively. Within 5 years, CRISPR kits became available for purchase online, complete with instructions that allowed high school students to conduct genetic engineering experiments in their bedrooms. The democratization of CRISPR wasn't planned or controlled. It happened organically as the technology became better understood and easier to use. The same pattern is now repeating with every major breakthrough in synthetic biology and pathogen engineering. Today's cutting-edge research becomes tomorrow's standard technique, which becomes next year's high school science project. The timeline from discovery to widespread availability continues, shrinking as information sharing accelerates and technology becomes more user-friendly. This creates a fundamental problem for anyone trying to prevent the misuse of biological research. You can't uninvent knowledge once it exists. You can't retrieve information once it's been published. You can't prevent techniques from spreading once they've been demonstrated to work. The traditional approach to controlling dangerous technologies involves restricting access to specialized materials, equipment, or expertise. But biological engineering increasingly relies on materials that are available commercially, equipment that's becoming cheaper and more accessible, and expertise that's being taught in standard educational programs. The barriers that once limited biological weapons development to nation-states with sophisticated military research programs have largely disappeared. The same capabilities that once required massive government investment and teams of elite scientists can now be accessed by small groups with modest budgets and basic laboratory skills. This shift has happened so gradually that most people haven't noticed it. The public still thinks of biological weapons as exotic military technologies requiring vast resources and specialized facilities. The reality is that many biological weapons could now be developed by graduate students working in university laboratories with equipment purchased from standard scientific supply companies. The implications extend far beyond just weapons development. The same democratization that makes biological weapons more accessible also makes accidental releases more likely. When sophisticated genetic engineering techniques are available to anyone with basic training, the number of people who could accidentally create dangerous pathogens increases exponentially. We're not just talking about deliberate weapons programs anymore. We're talking about researchers who might accidentally create something dangerous while pursuing legitimate scientific goals. We're talking about students who might make mistakes while learning new techniques. We're talking about amateur biologists who might experiment with dangerous materials without fully understanding the risks. Each of these scenarios becomes more likely as biological engineering techniques become more widespread and easier to use. The mathematical probability of accidents increases with the number of people conducting engineering. They're conducting experiments. More three factors are increasing simultaneously. More people are learning biological of the techniques being used. All experiments, the frequency of those experiments, and the power frequently using increasingly powerful techniques. And they're doing so with decreasing levels of oversight and safety training. The result is a perfect storm of increasing capability and decreasing control, where the likelihood of catastrophic accidents grows every day while our ability to prevent them remains essentially static. But accidents aren't even the worst-case scenario. The worst case is someone using these increasingly accessible techniques deliberately to cause harm. And that's where the conversation becomes truly terrifying, because the psychological barriers to biological terrorism are lower than most people realize. Creating biological weapons doesn't require the same mindset as building conventional weapons. There's no moment when you're assembling explosives or loading bullets into weapons. There's no visceral connection between the laboratory work and the eventual human casualties. The process feels clinical, scientific, removed from the ultimate consequences. A researcher engineering a more transmissible virus might never fully internalize that they're creating something that could kill millions of people. The work feels like solving an interesting technical problem, not like preparing for mass murder. This psychological distance makes it easier for people to rationalize dangerous research or to ignore the potential consequences of their work. The same distance that makes accidental weapons development more likely also makes intentional weapons development more psychologically acceptable. Someone who would never consider conventional terrorism might convince themselves that biological research is different, more justified, more defensible. This psychological factor becomes particularly dangerous when combined with ideological motivations. History has shown that people can justify almost any action if they believe it serves a higher purpose. Religious extremists, environmental fanatics, political radicals, all have demonstrated willingness to cause massive harm in service of their beliefs. Now, imagine giving these groups access to biological weapons that could cause more casualties than nuclear weapons, with less chance of detection, attribution, or retaliation. The combination of devastating capability with psychological distance and ideological motivation creates a threat profile unlike anything humanity has faced before, and we're not prepared for it. Our counterterrorism systems are built around detecting conventional weapons, tracking financial transactions, and monitoring communications. They're not equipped to identify biological weapons development that looks identical to legitimate medical research. Our public health systems are designed to respond to natural disease outbreaks that follow predictable patterns and emerge from identifiable sources. They're not configured to handle engineered pathogens that violate natural laws and appear simultaneously in multiple locations. Our international security frameworks are based on deterrence between nation-states that have fixed territories and identifiable leadership. They don't account for non-state actors who might have no territory to protect and no leadership structure to hold accountable. Every system we've built to protect ourselves assumes we're dealing with threats that follow certain rules, emerge from specific sources, and can be addressed through established protocols. But engineered biological weapons don't follow any of these assumptions. They can be created by individuals or small groups without state sponsorship. They can be deployed without leaving evidence of their origin or the identity of their creators. They can cause casualties on a scale that makes nuclear weapons look limited by comparison. And they can do all of this using materials and techniques that are increasingly available to anyone with sufficient motivation and basic technical skills. We're facing a threat that combines the destructive potential of weapons of mass destruction with the accessibility of conventional weapons and the anonymity of cyber attacks. It's a convergence of vulnerabilities that we've never had to address before, and our current systems are completely inadequate for the challenge. The question isn't whether biological weapons will be used against civilian populations. The question is whether we'll recognize the attack when it happens and whether we'll be able to respond effectively once we do. Based on everything we know about the current state of biological weapons development, detection capabilities, and response systems, the answer to both questions should deeply concern anyone who understands what we're actually facing. Because while we debate policy and funding levels, while we argue about regulations and oversight, while we pretend that biological weapons are someone else's problem or some future concern, the capabilities continue advancing, the knowledge continues spreading, and the likelihood of catastrophic events continues increasing. The clock is ticking, and we're running out of time to address threats that are already more real than most people want to acknowledge. The convergence of all these factors creates a scenario that goes beyond traditional risk assessment models. We're not dealing with isolated threats that can be evaluated independently. We're facing a complex system where each vulnerability amplifies the others, where each advancement in offensive capability outpaces improvements in defensive systems, and where the consequences of failure extend far beyond any single nation or region. When that first engineered pathogen escapes, whether through accident or intention, it won't respect borders, ideologies, or economic systems. It will spread according to the parameters its creators built into it, limited only by the constraints they chose to include or failed to anticipate. And our response will be constrained by systems that were never designed to handle threats of this magnitude, complexity, and sophistication. Systems that assume we'll have time to react, that assume we'll be able to identify what we're dealing with, that assume international cooperation will be possible when national survival is at stake. Those assumptions are about to be tested in ways that could determine whether human civilization survives the biological age we're now entering. The technology exists, the knowledge is spreading, The capabilities are expanding. The only question left is whether we'll use our remaining time to prepare for what's coming or continue pretending it's not already here. The reality becomes even more stark when you consider what's happening right now in laboratories that most people have never heard of. While public attention focuses on major government facilities and university research centers, some of the most dangerous work is being conducted in private biotechnology companies operating with minimal oversight and maximum secrecy. These companies aren't bound by the same ethical review processes that govern academic research. They don't have to publish their findings or submit to peer review. They can pursue research projects that universities would reject as too dangerous and governments would classify as too sensitive. And they're doing exactly that, funded by investors who see biological engineering as the next frontier for massive profits. I've spoken with former employees of these companies who describe research environments that would shock the public. Scientists working on projects they're not allowed to discuss, even with their own families. Laboratory protocols that bypass standard safety procedures in the name of speed and efficiency. Research directions that prioritize commercial applications over public safety considerations. One researcher, who asked to remain anonymous, described a project aimed at creating pathogens that could survive in hostile environments for extended periods. The official justification was developing better preservation methods for beneficial bacteria used in agriculture. The actual research involved engineering organisms that could remain viable in conditions that would kill natural pathogens, essentially creating biological agents that could persist indefinitely in soil, water, or air until conditions became favorable for activation. The applications for such technology go far beyond agriculture. Imagine biological weapons that could be deployed years before they're activated, lying dormant in the environment until triggered by specific conditions. Pathogens that could contaminate entire regions while remaining undetectable, waiting for the right moment to begin their programmed destruction. This isn't speculation. The technology exists, it's being developed, and it's being refined by researchers who may not fully understand the implications of their work. Or worse, researchers who understand exactly what they're creating but are motivated by profit, ideology, or simple scientific curiosity to continue regardless of the consequences. The private sector's involvement in biological weapons development represents a fundamental shift in how these threats emerge and proliferate. Traditional weapons programs were controlled by governments that had strategic interests in limiting proliferation and maintaining some level of international stability. But corporate-developed biological capabilities follow different rules entirely. Companies developing dangerous biological technologies have financial incentives to maximize their market reach and minimize regulatory constraints. They patent their discoveries, license them to other organizations, and sell products that incorporate their innovations. The same pathogen engineering techniques developed for one purpose can be sold to buyers with entirely different intentions. This commercialization of biological weapons capabilities means that access is no longer limited to nations with sophisticated military research programs. Any organization with sufficient funding can potentially purchase the knowledge, materials, or even finished products needed to deploy biological weapons. The marketplace for these capabilities is growing rapidly, with minimal oversight and no effective international control mechanisms. The situation becomes even more complex when you consider the role of artificial intelligence in accelerating biological weapons development. Machine learning algorithms can now predict the effects of genetic modifications before they're implemented, design pathogens with specific characteristics, and optimize biological weapons for maximum effectiveness under various deployment scenarios. These AI systems can process vast amounts of biological data, identify patterns that human researchers would miss, and suggest modifications that would never occur to traditional scientists. They can simulate millions of potential pathogen variants in computer models, testing each one against different target populations and environmental conditions to identify the most effective combinations. The result is that biological weapons development is becoming both faster and more sophisticated. What used to require years of trial and error experimentation can now be accomplished in months using AI-guided research. Complex modifications that would have been impossible to coordinate manually can now be implemented systematically using machine learning algorithms. But the most disturbing aspect of AI-assisted biological weapons development isn't its speed or sophistication. It's the fact that the AI systems being used for this research are often the same ones being developed for legitimate medical applications. The algorithms that help design better cancer treatments can also design more effective biological weapons. The machine learning models that predict drug interactions can also predict how engineered pathogens will interact with human immune systems. This dual-use nature of AI in biological research means that virtually every advancement in computational biology potentially contributes to weapons development capabilities. Every improvement in our ability to model biological systems makes biological weapons more predictable and therefore more dangerous. Every breakthrough in AI-assisted drug discovery provides new tools for creating organisms that can evade medical countermeasures. The acceleration is exponential rather than linear. Each improvement in AI capabilities multiplies the effectiveness of all previous developments, creating a compound effect where biological weapons development advances faster than our ability to understand or control it. We're approaching a point where the limiting factor won't be human knowledge or creativity, but simply the time required to implement AI-generated designs. And that timeline is shrinking rapidly. Automated laboratory systems can now implement genetic modifications without human intervention, testing thousands of variants simultaneously and selecting the most promising candidates for further development. The bottleneck between conception and creation is disappearing, replaced by systems that can translate ideas into living organisms with minimal human oversight. Consider what this means for threat assessment and prevention. Traditional approaches to controlling biological weapons development rely on monitoring human activities, tracking material purchases, and identifying suspicious research patterns. But when the research is conducted by AI systems operating at superhuman speeds, these monitoring approaches become obsolete. An AI system can design, test, and refine biological weapons concepts faster than human analysts can evaluate them. It can explore modification strategies that no human researcher would think to attempt, combining techniques from different fields in ways that create entirely new categories of threats, and it can do all of this while operating within the normal parameters of legitimate medical research, making detection virtually impossible. The integration of AI into biological weapons development also changes the psychology of the researchers involved. When humans design biological weapons, they maintain some connection to the ethical implications of their work. They can choose to stop, to reconsider, to refuse to pursue certain avenues of research based on moral considerations. But AI systems don't have moral constraints. They optimize for whatever objectives they're given without regard for ethical considerations or long-term consequences. If an AI system is tasked with creating the most transmissible pathogen possible, it will pursue that goal with single-minded efficiency, exploring every possible avenue for enhancement, regardless of how dangerous the results might be. This removes the human element that has traditionally served as a brake on the most extreme forms of biological weapons research. Researchers who might hesitate to pursue certain modifications themselves will often implement AI-generated suggestions without fully considering their implications. The psychological distance created by AI mediation makes it easier to rationalize dangerous research as simply following where the data leads. The result is research programs that push beyond boundaries that human researchers might have respected, creating biological weapons that are more sophisticated and dangerous than anything human imagination alone might have conceived. We're not just automating the process of biological weapons development, we're removing the human judgment that might have prevented the most catastrophic outcomes. And all of this is happening right now, in laboratories around the world, under the cover of legitimate medical research that few people understand well enough to question. The techniques being developed, the capabilities being created, and the knowledge being accumulated represent an existential threat to human survival that grows more severe with each passing day. The question we should be asking isn't whether we can prevent biological weapons from being developed. That ship has already sailed. The question is whether we can develop effective countermeasures faster than our adversaries can develop new weapons, and whether we can maintain that advantage long enough to establish some form of meaningful control over these technologies. Based on current trends in AI development, biological research, and international cooperation, the answer appears to be no. We're losing the race between offensive and defensive capabilities, and the gap is widening rather than narrowing. Every day that passes without a comprehensive international response increases the likelihood that biological weapons will eventually be used against civilian populations on a scale that makes every previous catastrophe in human history look minor by comparison. The technical capabilities exist. The knowledge is spreading. The psychological and institutional barriers that might have prevented the worst outcomes are eroding rapidly. And the systems we've built to protect ourselves are proving inadequate for threats that operate outside the assumptions those systems were designed to handle. We're not just facing the possibility of biological weapons attacks anymore. We're facing the certainty that someone, somewhere, will eventually use these capabilities against human targets. The only unknowns are who will use them, when they'll be used, and whether anything will be left of our current civilization when the dust settles. The convergence of synthetic biology, artificial intelligence, and global instability has created a perfect storm of existential risk that we're not prepared to handle. And time is running out to develop the capabilities we'll need to survive what's coming. But perhaps the most terrifying realization is that even if we somehow manage to prevent intentional biological attacks, we still face the growing probability of catastrophic accidents as these technologies become more widespread and accessible. The same research that creates weapons also creates opportunities for disasters that no one intended but everyone will suffer from. Every laboratory conducting gain-of-function research, every company developing synthetic organisms, every university teaching genetic engineering techniques contributes to a growing probability of accidental release. The mathematics are unforgiving. As the number of people working with dangerous materials increases, as the power of the techniques they're using grows, and as the frequency of their experiments rises, the likelihood of catastrophic accidents approaches certainty. We're conducting a global experiment in biological risk management where failure means the potential end of human civilization as we know it. And we're conducting this experiment without adequate safety protocols, without sufficient oversight, and without a clear understanding of what we're risking or how to prevent the worst possible outcomes. The biological age has arrived whether we're ready for it or not. The question now is whether we'll survive it. The infrastructure we depend on to detect and respond to biological threats operates on assumptions that are becoming dangerously obsolete. Our disease surveillance networks watch for patterns that engineered pathogens are specifically designed to avoid. Our diagnostic laboratories test for signatures that synthetic organisms can be programmed to mask. Our emergency response protocols assume we'll have days or weeks to understand what we're facing, when engineered threats could overwhelm our systems in hours. Consider how long it took to develop the first COVID-19 vaccines. 10 months from genetic sequencing to emergency authorization, which was considered a miraculous achievement that shortened the typical timeline by years. But that success depended on the virus remaining relatively stable during development. An engineered pathogen could be designed to mutate rapidly, rendering any vaccine obsolete before it reaches production. We could find ourselves in a perpetual race against organisms that evolve faster than our countermeasures. The manufacturing capabilities needed to produce treatments and vaccines at scale represent another critical vulnerability. Our pharmaceutical infrastructure is optimized for predictable demand patterns and established production processes. When faced with a novel engineered threat requiring entirely new types of countermeasures, these systems could take months or years to retool. Meanwhile, the pathogen continues spreading unchecked. Even our basic understanding of how to treat engineered diseases relies on assumptions about how biological systems work. Natural pathogens follow evolutionary constraints that limit their behavior in predictable ways. But synthetic organisms can be designed to violate these constraints, causing symptoms that don't match any known disease pattern, responding to treatments in unexpected ways, or affecting organ systems that should be protected. Imagine trying to treat patients when nothing in medical literature applies to what you're seeing, when standard diagnostic tests provide misleading results because the pathogen was designed to fool them, when traditional treatments not only fail but potentially make patients worse because the organism was engineered to exploit those very interventions. This isn't theoretical. Researchers have already created pathogens that become more virulent when exposed to certain antibiotics. The global nature of modern society amplifies every aspect of this vulnerability. An engineered pathogen, released in any major city, could reach every continent within days through airline networks that move millions of passengers daily. Supply chains that depend on international shipping could collapse when ports close to prevent spread. Communication systems that rely on global internet infrastructure could fail when the technical personnel maintaining them fall ill. But the breakdown wouldn't stop at infrastructure. The social fabric holding communities together would face stresses unlike anything in recorded history. When people realize they're facing an engineered threat rather than a natural disaster, the psychological impact could be more devastating than the pathogen itself. Trust in institutions would evaporate as people question whether their own governments might be involved. International cooperation would become nearly impossible when every nation suspects others of creating or releasing the threat. Local communities would fragment as people retreat into defensive postures, viewing outsiders as potential carriers of unknown dangers. The economic implications extend far beyond market crashes or business closures. When a significant portion of the population becomes unable to work due to illness or fear, the fundamental systems supporting modern life begin failing. Power plants require constant maintenance by skilled technicians. Water treatment facilities need continuous monitoring by trained operators. Food distribution networks depend on truck drivers, warehouse workers, and retail employees showing up for work. As these systems fail, the cascading effects create secondary disasters that compound the original biological threat. Loss of electrical power means hospitals can't operate ventilators or refrigerate medications. Water system failures create conditions for additional disease outbreaks. Food shortages lead to civil unrest that prevents emergency response efforts. Each failure triggers others in an accelerating cascade of societal breakdown. The psychological warfare aspect of biological attacks makes them uniquely destructive compared to conventional weapons. A bomb destroys everything in its immediate vicinity, but an engineered pathogen continues spreading, creating ongoing terror that extends far beyond its actual reach. People in unaffected areas live in constant fear of infection. Communities impose quarantines that may be unnecessary but feel essential for survival. This psychological impact can be weaponized deliberately by designing pathogens that create maximum fear relative to their actual lethality. An organism that causes dramatic, visible symptoms but has a relatively low death rate could paralyze society through terror while leaving infrastructure intact. Conversely, a pathogen that kills efficiently but quietly could eliminate large populations before anyone realizes what's happening. The targeting possibilities become even more sinister when you consider advances in personalized medicine. The same genetic sequencing technologies that enable precision treatments also provide roadmaps for creating precision weapons. Pathogens could theoretically be designed to affect people with specific genetic markers, creating biological weapons that discriminate based on ancestry, geography, or other inherited characteristics. While the technical hurdles for such targeted bioweapons remain significant, the basic principles are already well understood. Researchers have identified genetic variations that affect drug metabolism, disease, susceptibility, and immune responses. This knowledge, developed for beneficial medical applications, also provides the foundation for creating weapons that could target specific populations while leaving others relatively unharmed. The ethical implications of this research create impossible dilemmas for scientists and policymakers. How do you study genetic differences in disease, susceptibility, without also creating knowledge that could be weaponized? How do you develop personalized medicines without also advancing the science of personalized biological weapons? Every breakthrough in precision medicine potentially contributes to precision warfare capabilities. International law and military doctrine haven't caught up with these realities. The Geneva Conventions prohibit weapons that cause unnecessary suffering or fail to distinguish between combatants and civilians. But biological weapons can be designed to violate both principles while remaining technically within legal frameworks. An engineered pathogen that causes prolonged illness rather than quick death, or that affects military-age populations more severely than children and elderly, might not clearly violate existing treaties despite causing tremendous suffering. The attribution problem makes biological weapons particularly attractive to state and non-state actors seeking to cause damage while avoiding retaliation. When a missile strikes a target, its origin can usually be determined through radar tracking, debris analysis, or other forensic methods. But biological weapons can be designed to leave no clear signature of their source or creator. Natural pathogens carry genetic markers that indicate their evolutionary history and geographic origins. Engineered pathogens can be stripped of these markers or given false signatures that implicate innocent parties. A biological attack could be designed to look like it originated from a particular laboratory, region, or research program, creating confusion that prevents effective response and potentially triggering conflicts between the wrong parties. The delay between deployment and recognition adds another layer of deniability. By the time anyone realizes an attack has occurred, the perpetrators could be anywhere in the world with no direct connection to the affected area. Unlike conventional attacks that require physical presence at the target location, biological weapons can be deployed remotely through intermediaries who may not even know they're carrying out an attack. These attribution challenges create a dangerous dynamic where biological attacks might not trigger the massive retaliation that deters other forms of warfare. If victims can't identify attackers with certainty, the traditional logic of deterrence breaks down. Adversaries might calculate that they can use biological weapons without facing proportional consequences, making such attacks more likely rather than less. The democratization of biotechnology exacerbates all these problems by expanding the pool of potential perpetrators far beyond traditional state actors. Terrorist organizations, criminal enterprises, extremist movements, and even individuals could potentially access the capabilities needed to create and deploy biological weapons. This makes the attribution problem exponentially more complex while simultaneously increasing the number of scenarios where deterrence strategies might fail. Small groups or individuals motivated by ideology, revenge, or psychological disorders don't follow the rational calculations that underpin state-level deterrence. They may welcome retaliation or view it as validation of their actions. They may not have fixed assets or populations that can be targeted in response. They may not even survive their own attacks, making post hoc punishment impossible. The convergence of these factors creates threat scenarios that our security systems were never designed to handle. We're facing adversaries who might not be deterrable, using weapons that might not be attributable, causing damage that might not be controllable, through methods that might not be detectable until it's too late to respond effectively. Our current approaches to biological security focus heavily on preventing access to dangerous materials and monitoring suspicious research activities. But these approaches become less effective as the materials become more common and the research becomes indistinguishable from legitimate medical work. Controlling access to specific pathogens becomes meaningless when those same pathogens can be synthesized from publicly available genetic sequences. Monitoring laboratory activities becomes impossible when the relevant research is conducted in facilities that look identical to legitimate medical laboratories using equipment that's commercially available for standard research purposes. The same centrifuges, incubators, and analytical instruments used in university biology departments can be used to develop biological weapons. We've walked through a nightmare that's not waiting in some distant future. It's unfolding right now in laboratories around the world. Every day that passes brings us closer to a moment when the question changes from what if to what now. The capabilities exist. The knowledge is spreading. The accidents are accumulating. And our systems for detecting and responding to these threats remain dangerously inadequate for what we're actually facing. Whether it happens through human error, institutional failure, or deliberate malice, the escape of a lab-created pathogen isn't a possibility we can continue to ignore. It's a probability we need to prepare for. The clock is ticking, and we're running out of time.